So what we've got here is the thylakoid membrane. So this is inside the thylakoid at the top, and this is outside the stroma. So what happens is the first thing is light strikes the chlorophyll here, and an electron is excited and liberated. It gains energy and tries to zing off into the universe, but it's captured by the electron carrier here. So it captures it and will move it forward. These are all electron carriers. They're proteins which can capture electrons. This is a little bit like heating up a potato for 20 minutes in the microwave and then throwing it at someone. They're going to immediately drop it. It's too hot. But if you say they can't drop it, they're going to juggle it in their hands for a few moments and then they're going to have to pass it to someone else. So this is exactly what happens. The hot potato of the electron is passed through the carrier chain, moving through and ultimately ending up in this molecule here. But we're going to leave it for a little while. Now, what happens is hydrogen ions, which are currently within the thylakoid in the form of water, are going to be split by the light as well. So what happens is we've got water up here, light strikes the water and splits it into oxygen, which is a waste product. So that's going to leave the leaf, hydrogen ions, and an electron. Because the chlorophyll here has lost an electron, it now gains an electron. So we've now stabilized the chlorophyll and it's returned back to a happy state where it's got its electron back. So it's now fully stabilized again. That electron, as it moves through the chain, has the interesting effect of being able to pump hydrogen ions via active transport across the membrane. This allows hydrogen ions to build up on the inside of the thylakoid. We'll end up with many hydrogen ions building up there. Water is being split constantly by light, and also hydrogen ions are being pumped across the membrane, which means we get a build-up of hydrogen ions on the inside of the thylakoid. This is a little bit of a mind-blowing moment for most people because they've learned that for active transport to occur, it requires ATP. This is one of the cases where ATP is not used, is the energy from the electron that pumps the hydrogen across. We've now built up hydrogen ions on the inside of the thylakoid within the space there. They are now going to diffuse via a process called the chemiosmotic theory. It's kind of like chemical osmosis through this molecule here. This molecule is ATP synthase, sometimes referred to as ATPase. And it is a fascinating molecule, which we'll look at in a little bit more detail in a bit. And as the hydrogen ions move through the ATP synthase, this forward and backwards facing capital D here, these subunits spin and ADP and a phosphate ion are combined to form ATP, our high energy unit. That's the first thing we want to make. That's then going to go off and assist with the other cycle, which we're going to look at later. So we end up with a buildup of hydrogen ions on this side. Most of these get pumped back across the membrane through the energy of the electron there. So most of them go, but we do end up with a buildup of electrons here and hydrogen ions here. This is where oxidized NADP comes from the cycle, which we can find out about later, and arrives here. It's now going to take the electron and the hydrogen ion, and it's going to become reduced NAD, sometimes referred to as NADPH. We have now made our two units we want to make. We have made oxygen as a waste product at the top. We have made ATP energy unit, and we've now got a reducing agent, which can now go off to power the next cycle, the Calvin cycle, which we're going to look at later. So that process of hydrogen ions moving across the membrane is called the chemiosmotic theory, chemical osmosis, and it's theoretical because they haven't fully proved it yet. It involves the movement of ions across a partially permeable or selectively permeable membrane down a concentration gradient exactly the same way as osmosis does. And what happens is ATP is generated by those hydrogen ions moving through the ATP synthase. This means that high energy units can be made relatively simply. And because it's moving across a partially permeable membrane down a concentration gradient, it's kind of similar to osmosis. So they've called it the chemiosmotic theory. So chemical osmosis. ATP synthase is an amazing molecule. Look into it in more detail. Trust me, it's a fascinating little molecule. It consists of an axle and 6 subunits that can spin. I like to think of it as a little bit like a water wheel. So what happens is the hydrogen ions can move in through the top here. So they'll move in through the top here through this channel. And as they go, this subunit will spin. So there's a channel here, and it's held in place by this bar here. But as it moves through, this section here, the F1 section, spins, and ATP is synthesized from ADP and a phosphate ion. It's a fascinating molecule. And as you can see here, it also is found in the mitochondria. It is a very complex process, photosynthesis. This section is incredibly complex. There's a few diagrams that you can help with this that can build upon it. You've got cytochrome, you've got ferredoxin complex, loads of different molecules that consist of this. Luckily, at this level, we don't need to know it, but it's really good for synoptic assay links later, so please look them up in your own time. There's definitely many more molecules involved that we talk about, and it gets very, very complex. But the key two, which you can look up, are cytochrome and ferredoxin complex. They're both part of those molecules. So it's just giving a little bit of a name to those molecules. Just so instead of calling them molecules, we can actually give them a structure and a name. Final few bits. The first thing is photolysis, the photolysis of water, or breaking using light. So the formula is there. We need to know this. So it's a balanced formula. It's not too difficult to remember. You've got 2 water and it becomes 4 of each, apart from the oxygen. And all it is water is split into protons, electrons, and oxygen using light. And the thylakoids themselves are arranged in such a way that they maximize the light capturing chances. So they've got a large surface area, they're held in place and angled towards the sun. There's also lots of DNA and ribosomes in the chloroplast to aid protein synthesis, because this uses an essential enzyme, rubisco, which is vital for this to work. We're going to encounter rubisco later when we get onto the Calvin cycle. I also mentioned the photosystems. We have 2 photosystems. The first one is photosystem 2. It was discovered after photosystem 1, hence its name, but that is the absorption of light, electrons being excited, and photolysis. Photosystem 1 was encountered first, it actually occurs after, and it involves the manufacturing of NADPH, or reduced NADP, and ATP. You don't really need to know these in depth, just have an awareness, because I've seen a few exam questions that have them on there. They are quite easily split, but it is very difficult to get your head around that photosystem 2 happens first, photosystem 1 comes after. It's just because they were discovered in a different order. So it's the oxygen and electrons, and it's the production of NADPH and ATP. And final thing is just the structure of NADP itself. It's a very complex molecule. I often get asked what it stands for. There it is down the side there. The key bit is the bit at the top, the bit where you've got the site of the hydrogen atom additional attachment, the positive charge. That allows it to loosely carry a hydrogen ion. So when it picks up that hydrogen and becomes NADPH, it can carry that hydrogen loosely for a period of time and then give it to another molecule. And that's the key bit of this. It's a hydrogen acceptor which carries the hydrogen for a brief moment in time and then gives it to another molecule to help the reduction of CO2 into glucose. Thanks a lot. Bye.